nothing here.
He's being kept track of.
Housed in one of the last remaining buildings of the Navy's World War II torpedo testing range, the East Hampton Town Shellfish Hatchery is located on the shore of Fort Pond Bay in Montauk. The day starts with a trip to the pump house to begin the flow of water to the hatchery. Water entering the hatchery passes through filter bags, or socks, which catch debris and undesirable plankton, the small plants and animals present in raw seawater. A large tank receives the filtered water, and here it can be heated to any desired temperature. Since the receiving tank is on the second level, water may be gravity fed to other tanks on the hatchery floor below. First, this filtered seawater is used in the production of algae, which is food for the shellfish.
These different colored flasks represent different algae that we maintain in the algae room. These smaller vessels are used to make larger vessels, such as 5-gallon carboys, 50-gallon tubes, or 1,000-gallon mass culture tanks, which are then fed out to the shellfish.
Algae is the common term for microscopic plant life, also known as phytoplankton. Many different species of algae have been found by scientists to be particularly good shellfish food. The best have been isolated, or singled out from seawater to be grown in pure or xenic cultures, consisting of only one species. A specialized marine plant food, or media, consisting of nutrients and certain minerals and trace elements, is added to sterilized seawater as a first step in growing the algae. Stock algae cultures are maintained at the hatchery year-round. Each week, a new stock culture is made by transferring a small amount of a mature culture into a flask containing the sterile seawater and nutrient media. To avoid contamination of the cultures, the transfer process takes place in a bacteria-free environment under a transfer hood bathed with ultraviolet light. A cotton and cheesecloth stopper is fitted to the neck of the flask in order to keep the contents clean, yet allow some air exchange, a necessary element in plant growth. The flask lip is flamed before and after removing the stopper in order to kill bacteria lodged there. Two types of algae are grown in the hatchery: flagellates and diatoms. Flagellates, the smaller of the two, have thin cell walls and a whip-like tail structure called a flagella, which gives the cell mobility. Flagellates are used to feed the shellfish in their early stages and as a diet supplement later on. Diatoms are larger than flagellates and have beautifully formed crystalline cell walls made largely of silica, a common earth mineral. Diatoms are fed to the shellfish in their later stages of growth. Algae is cultured in ever larger quantities, each stage requiring about a week to mature to a greenish golden color, indicating the desired cell density of about 1 million cells per milliliter, or less than a drop, of seawater. Every day, about 600 gallons of algae culture is used for feed, and its growing process must be continually repeated to meet the demand. Shellfish used for breeding, called the broodstock, is gathered shortly after the new year. Pulling broodstock out of the water after having been overwintered. Broodstocks are breeding stock. These are clams and oysters. We pull scallops out later. We'll be bringing them to the hatchery to get ready to spawn. For about 6 weeks, they are conditioned to spawn by being held in 65-degree water and constantly fed with algae. These conditions simulate late springtime in the harbors, allowing the gonads, or reproductive organs, to develop. Spawning, the release of sperm and eggs into the water, can then be triggered by a simple 10 or 15-degree rise in water temperature, imitating summer conditions. Mass spawning, where eggs and sperm of many animals are allowed to intermingle, allows several desirable genetic characteristics to be bred into the resulting batch of offspring, such as fast growth, local vigor, and disease resistance. Sometimes, a little sperm dripped near the siphons of uncooperative breeders will spark them into action. Fertilization of the egg by the sperm takes place in the water, but care must be taken not to allow too much sperm to mix with the eggs, or poor development will result. So males are removed from the tank as they begin to release their sperm. While oysters and clams are either male or female, bay scallops can produce both eggs and sperm and are called hermaphrodites. Each spawn can produce as many as 200 million fertilized eggs, and these are placed in tanks called conicals for further development in the warm larval culture room. After fertilization, the egg divides rapidly until in just a few hours a swimming form called a larvae results. Although there is more than one larval stage, hair-like cilia called the velum, which allow larvae to swim and collect food, gives rise to the name of the most common larval form, the veliger. Water is changed regularly in the conicals, with the larvae being caught on fine screens and graded to size through a series of sieves. The larger animals returned and the runts discarded. In about a month, larvae metamorphose, or begin to change into bottom dwellers, losing the velum and developing an active foot, a transition called the pediveliger stage, and often referred to as setting. The setting animals will adhere to tank surfaces, clams and scallops with a thread-like structure called the byssal thread, which can easily be dislodged with a fine spray of water. Oysters must be provided with a fine bed of shell bits called culch on which to set. Individual oysters cement themselves to the tiny bits of culch, which can then be easily handled. Shellfish are held in shallow set tanks in the warm larval culture room for about 2 weeks after set, until they are about 1 millimeter in length, or a little over a 1/16 of an inch. In their final 2 to 4 weeks in the hatchery, shellfish are moved to a separate room and placed in downweller tanks for additional growth. Downwellers are tubs fitted on one end with fine screen. Recently set animals are placed on the screen, and tank water laden with algae is recirculated through the bed of shellfish in a downward motion. As with all hatchery systems, a regular takedown schedule consisting of draining, rinsing, refilling, and feeding must be maintained. By allowing water temperatures to slowly drop, this stage also acts to acclimate the small shellfish to conditions they will encounter in the nursery system, their next destination. In springtime, estuaries provide young growing shellfish with an unrivaled, abundantly rich mixture of algae and other microscopic nutrients, ideal for growth. As soon as harbor and bay waters warm, juvenile shellfish are moved to a nursery on 3 Mile Harbor in East Hampton, where the fertile water is pumped through upwellers in tanks on shore. Like downwellers, these are also tubs screened at one end, but the harbor water pumped into the tank now passes up through the bed of shellfish on the upweller screen. When conditions are right, the volume of shellfish can double in just a few days. To assure the optimum growth and continued good health of the animals, constant attention must be paid to keeping the system clean and the shellfish graded to size. Shellfish that were just 2 or 3 millimeters when they arrived in the nursery leave at about the size of a small pea.
Sieving has a couple purposes. Cleans them up, separates the ones that are growing faster from the slower growers, and then allows us to have the room to move the larger ones to the field system, and then we just put the smaller ones back here.
As summer arrives and the shellfish get larger, limited land space and the high cost of pumping water compel the practical grower to turn to a field grow-out system for what will amount to the longest and most labor-intensive part of the growing season. What started out in the hatchery as a mere thimbleful of fertilized eggs, by the end of the growing season will result in tens of thousands of pounds of seed shellfish. The hatchery's field site is in Nalepinski Harbor in Amagansett, an exceptionally pristine and naturally productive water body. The site's proximity to an inlet to the bay ensures the good water exchange needed to secure the growth of millions of seed shellfish. Depending on the species, a variety of screen trays and nets are employed in the process, but the mainstay of production centers around stacked tray blocks suspended in floating rafts. Each day, tray blocks are removed from the water, where the seed shellfish are taken out and graded to size.
Sieving the oysters, and the ones that go through the holes obviously are smaller, and then we put them on a different screen. The bigger ones go on a larger screen once they...
Wet blocks, usually beginning to foul with a variety of marine settling organisms, are rotated to a drying area nearby. It is important to return to service each block at least every 2 weeks. Otherwise, uneven growth of shellfish in the trays will hinder their development, and fouling organisms will become a problem, impeding water flow through the stack of trays. Shellfish requiring further growth are restocked into clean, dry blocks and returned to the rafts for further growth. If large enough, the shellfish will be seeded or planted on public grounds in one of the town's harbors, or in the case of some of the clams, brought to a site in which they will be held over the upcoming winter for additional growth the following year. This clam overwintering takes place in Fresh Pond, Amagansett, and is done with the assistance of some of the baymen familiar with conditions in the area.
Well, I'm glad to hear it growing. Now, John?
Well, I was pretty surprised, you know, because we never saw this good a survival when we'd done this in the past here.
Right. The ones you put in earlier grew like a sunflower. They're that big already.
Seed clams are placed in shellfish growing bags and strung along the bottom of the pond on long lines, making retrieval easier the following fall. The mesh of the bag affords the clams inside protection from their enemies while allowing some of the bottom mud to sip through into the bottom of the bag. It's very important to the clam to be able to burrow into some bottom sediment by the time it faces its first winter. Some shellfish seed is also provided to the New York State Department of Environmental Conservation for stocking state public grounds adjacent to the town. This is in exchange for the state having given East Hampton a grant with which to begin its hatchery.
Hi, my name is Joshua Teal. I represent the New York State Department of Environmental Conservation. I'm a biologist there and I work under shellfish management. Today we are seeding hard clams and oysters as part of a joint agreement with East Hampton Town Hatchery. Basically, as part of an original agreement that helped fund this hatchery and all the work therein, the state is allocated 10% of the total production of the year. We take that production and then we seed it out into local state waters for the benefit of local baymen and private individuals. The East Hampton Hatchery is very well run. They have a very high production for what they do, very high quality. They're very professional in how they go about things.
See you later.
Safe trip.
Testing the effectiveness of planting seed shellfish to enhance the natural resource is an important part of the hatchery's work. The cooperation of other agencies and educational institutions can carry mutual benefits in this respect. Recent work in Accabonac Creek is a good example.
Hi, I'm Steve Tettleback. I'm a professor at Southampton College of Long Island University. We're out here today working with the town of East Hampton Shellfish Hatchery to assess the success of their clam plantings. This is something we've been doing now for a number of years, and it's a great benefit to our class. We get students out in the field here, in the water, they're picking through, they're getting to see what it's all about in terms of raising clams and planting them out and trying to enhance the resource of the town. We've worked with John and Craig and Frank for a number of years now, and this has actually been kind of a springboard for a number of our students, because we've had students, for example, Beth over here took my class 2 years ago, and she's now working with the town, and so this has been a real good learning experience for our students, and gets them to see what goes on in the real world, and of course that's what it's all about. So
The Narrated clams, which are provided by the hatchery, are being kept track of, as well as the white clams, which would be found as a natural stock in the area that's being suction dredged. We're here to, what we did, we seeded a plot this morning with Narrated clams as well as some stones. So the kids from Southampton College are out there suction dredging.
That's running a pump. It pumps the air right through here. And basically, she takes this hose, puts it in the ground, and sucks up whatever's there, clams, rocks, shells, anything. And it runs right through. We have a bag back here, and it holds it all. And once we're done, take this off, in a bucket, pour it into a bucket, take it over there, and people sort it.
They're the red ones.
They're the red ones.
The hatchery spearheaded an important experiment also being conducted in several other Long Island towns, which is testing the relative survival of seed clams planted at different sizes and in different months of the year in 9 1/4 acre plots. In East Hampton, this work is being carried out in Northwest Creek.
We have 22,500 clams for each of these plots. So we're just measuring them out into 5 portions. And then a person will take each bucket and walk between 2 sets of poles and throw the clams out or seed them out. So.
Later in the fall, clams held since last year in Fresh Pond are retrieved. Bigger now, perhaps better able to fend for themselves.
The biggest problem with growing shellfish from the first year to harvest, which is about another 3 to 5 years, is that there's a lot of predation over that period of time, primarily by crabs. Blue claw crabs, green crabs, mud crabs, small mud crabs in the case of small clams. So these predation problems are the reason that seed clams have a fairly low survival when put into wild conditions. What we're doing here in Fresh Pond is culturing clams through a second season in order to get them a little bit bigger before we seed them into the wild. And we expect that this would have the result of increasing the chances of these clams surviving to harvest size.
These hard clams in this tote here are from this season. They're about 6 months old. They've been growing out in our field gear. They're pretty small still. And these in this tote have been out in the bags in the Fresh Pond for about a year. They went in at this size, and now this is about a year's growth here. You can see the difference. These small ones are going to be put into the bags today, and we're going to grow them for a year.
As the weather deteriorates, some last-minute work needs to be finished up at the seed planting experiment.
We're here at Northwest Creek and we're doing a clam seeding project that's taking place in a couple of different areas along Long Island. And here today we're suction dredging for clams that we've seeded in previous months throughout the summer. So we have 2 teams now out there suction dredging. We have 6 plots to dredge today, and hopefully we'll find some clams from previous seedings.
With falling water temperature, growth of the shellfish slows, and seed remaining in the grow-out systems must be released into the wild. It is especially important to release clams before temperatures drop below 50 degrees, or they might have trouble burrowing into the bottom and fall easy prey to predators. After the devastating brown tides of the 1980s and 90s, bringing back the once vibrant bay scallop is a priority.
We have here bay scallops, approximately 8,000. They were spawned 5 months ago, and they've reached about 35, 40, some 45 millimeters total length. So what we'll do is we'll take them out and seed them in the harbor today. Hopefully they survive and grow, be harvestable in a year. Here we have 70,000 hard clams. These are approximately 6 months old. These 2 are about this size, 15, I'd say anywhere from 14 to 16 millimeters in size that we'll seed out at the end of the year. It's our final grow out size. And these oysters here, approximately 60,000 oysters were spawned 8 months ago. And they're anywhere from, I'd say, 20 to 30 millimeters in size. And these also are about the size that we seeded out today.
A sample count is used to track the distribution of shellfish throughout town waters.
Hans Creek here has had some eelgrass and scallops in the past, so we think it's a good nursery. So we've seeded this in the past, done some overwintering here, and had some pretty good success. Our theory is if they set up in the creek here, they'll be flushed out of the inlet over time and repopulate outside the main harbor. That's our hope.
probably the coldest and most delicious clams we've ever had. They came from Lake Montauk. They're opening up nice. And if you look at this, wouldn't you want to eat one of those?
Is doing wonderful things. The clams, I think, are very important to this whole area, to the northern part of the United States. And it keeps us closer to the land. And if you go clamming barefoot, there's nothing like that.